system. So bladder, urethras, ureters. I'm thinking about all of the blood vessels in your body, all of the lymphatic vessels in your body. We're talking about a lot of hollow things. If you look at the anatomy of those structures, you will find epithelial linings. So we've talked about the epithelium. You have some impression already about transitions and the type of epithelium in both the respiratory and digestive system. You were supposed to pick that up as we went. So if I said, tell me about the transitions of epithelium as you're working your way through the respiratory and digestive system, you should have some idea already about what's happening there. There's logic behind that. The middle layer of these hollow things that we find in the body is smooth muscle. And most of it's this stuff right here. These spindle-shaped cells, they're single unit, they're connected by gap junctions, they behave as syncytiums, they have varicosities innervate them. Very common. I want to talk to you about that today. This kind of smooth muscle. Now, please don't oversimplify it. I have a tendency to do that as a teacher because when we get to the digestive system, I'll ask you this again. Tell me about the wall because we have to learn the walls, what they look like. And you're going to say, you told us very early on, it's epithelium on the inside, smooth muscle in the middle, and then a connective tissue covering on the outside. That's what you told us. And that's true. But we're going to see variations in that. So if you just, back off of it and look at it big picture, yes, there's epithelium there, there's smooth muscle there, and there's peritoneum, visceral peritoneum, connective tissue there. So yes, that's true, but there are going to be nuances to that. I'm saying this is a general feature, so I'll point those out to you as we get to each of them. Now, I also like to introduce here, although normally I just say it and we'll show the picture later, but today I'm going to show you the picture. I put it in here before class because I'll just compare. We didn't do all the muscle in one lecture period, so this gives me an opportunity to do a little review with you. So up here I've got a picture of what we call multi-unit smooth muscle, and here single unit. Now the picture you saw on the previous page with these spindle-shaped cells is an example of what we call single unit smooth muscles. This is the majority of the smooth muscle you find in your body. So this is the smooth muscle, for example, you find in the walls of the blood vessels. This is the smooth muscle you find in the walls of the respiratory, the smooth muscle in the walls of the digestive system, the smooth muscle in the walls of the uterus, the smooth muscles in the walls of the bladder. So this is most of the smooth muscle in the body is of this variety. And there are some anatomical features that explain some things about this kind of smooth muscle. First thing you notice is it is typical to find gap junctions between the smooth muscle cells. Now, where have we seen that before? Where have we seen gap junctions between cells before already?
Is it the cardiac muscle?
Mm-hmm. In the heart muscle. So I made this case to you last time we were in class together that cardiac muscle normally behaves as a syncytium, a single unit, because the cells are connected by these gap junctions. So it's not going to surprise you to learn later about the functional capacity of these guys working in conjunction with each other. All right? Now, I didn't further emphasize with the heart muscle that oftentimes when you find layers, cellular layers that are connected by gap junctions, they have unusual types of innervation. And the innervation of those areas, the innervation patterns are unusual apparently because of the ability of those cells to connect to each other. In other words, you don't really need to stimulate each individual cell here if you want the entire mass to contract. So that's the way it works with your heart. All you do is start the signal in the heart and it spreads from cell to cell. You don't have to have a neuron, an innervation piece going to each cell. So that's true in heart. That's true also in these syncytia. or singular syncytium that exist within our digestive and respiratory systems and urogenital systems, blood vessels, we find that these guys tend to contract as units, whole sheets contracting. Now, the unusual anatomy for the innervation is, you can see illustrated here in a very sort of dumbed down way. You can see here that the innervation from the autonomic nervous system here are called autonomic neuron varicosities. Now, in order to understand what we mean in anatomy by varicosity, let me show you the difference in the way skeletal muscle gets its information. So, in both single and multi-unit smooth muscle, you can see the varicosity here associated with the cellular, with the layer of cells. In the multi-unit, you can see there appears to be more specificity. Can you see that? The autonomic innervation gets closer to more cells in a multi-unit smooth muscle, which means you're going to have finer control of multi-unit smooth muscle than you are of single-unit smooth muscle. So this stuff works in sheets. This stuff works more like individual cells. You have more control over the multi-unit smooth muscles. Let me give you a couple examples. In the next chapter we do, it's coming up in this presentation, we're going to look at the anatomy of skin. And one of the things we're going to see in skin is the hair follicles that are all epidermal derivatives are pressed down into the dermis. And as such, in the dermis, they are connected to muscles that are anchored within the dense irregular connective tissue. Muscle attachments that look like a regular muscle that goes from here to there. But they're attached to the hair follicle. And I'm going to tell you when we get there, they receive autonomic varicosities. They are innervated by the sympathetic nervous system. And when those muscles are stimulated, they contract in much the way we would envision a regular skeletal muscle would contract. And when they do, they cause the hair follicle to stand up. And for that reason, they're called erector pili muscles. They cause it to erect or stand up. And so where you have hair follicles that haven't broken through, it causes little dimples on the skin when that happens. We call them goosebumps. Those are multi-unit smooth muscle cells. There's another group of them in our eyes that actually help to control the shape of the lens in your eyes. And so this will come later in this semester as well, and I'll show you these muscles. We say to ourselves, as we're reading, we say, well, I'm reading and my eyes are tired. What kind of fatigue is that? Well, it's muscle fatigue. But most of the time people think about muscle fatigue in relationship to skeletal muscle. Muscles are contracting, I wear them down, and I have muscle fatigue. These are actually multi-unit smooth muscles in your eyes that are behaving much like skeletal. They have much finer control. There are a few gap junctions not shown here occasionally that show up in them, but for the most part you don't see the gap junctions. So when you have the sheet contracting, it sometimes can make itself go, but oftentimes what happens is the chemicals that are released from the nervous system hit the top layer. So you can see just laid across the top of the sheet here, and then the signal spreads through the sheet by way of gap junctions. Very interesting control of smooth muscle. Let's compare that to the skeletal. So look over here, you can see a skeletal muscle fiber here, and you can see the connection from the nervous system to the skeletal muscle fiber is a very different arrangement. Notice that it appears that the end of the motor neuron, the connector to the skeletal muscle, is almost like it is plugged in to the muscle. And that's a good way to envision it. It's not actually touching the cell, but it does look as though it's plugged in. The control of skeletal muscle cells is exquisitely specific. And if you're going to make a muscle contract that's skeletal, you're going to talk about motor units. Individual cells that are stimulated to make the contraction stronger. So very specific connections in the nervous system for skeletal. Varicosities rule the day for smooth muscle and cardiac. And you can see now the differences between the multi and single smooth muscle. So you realize now why I didn't have time to do this with you on Wednesday. So I'm going to emphasize this to you one more time before we leave. It's very important anatomy. It will speak to our physiology next semester. We have much to say about what happens when those little purple dots come out of those varicosities. We have much to say about what happens when these little green dots come out of their thing and cross the synapse. This is where physiology begins, but you got to learn the anatomy first. So this is smooth muscle, and I've compared it for you with cardiac and skeletal. All right, the last tissue is nervous tissue, and nervous tissue is made up principally of two general cell types, neurons and glial cells. The neurons of the nervous tissue are what we call the conducting cells. So whatever we say about the physiology of the nervous system in this class, whatever we say, you're going to have to learn the pathways to muscles. So if somebody says to you, that they have weakness in the lateral forearm, then you're going to be able to tell someone what nerve is a problem and how you would get back to the spinal cord on that nerve. So that's coming in this class, learning those things. So we're talking about either sensory or motor stuff as it pertains to the nervous system in this class. We're going to be talking about neurons. So these are the conducting cells carrying information away from and to the central nervous system. And there are varieties of these neurons. They're typically listed in most texts 3 different varieties. And when I get to the nervous system, I'll show you each and give you examples for where they are. Generally speaking, a neuron has a cell body, sometimes called a perikaryon, and it will have one or multiple dendritic sensory information gathering pieces attached to the cell body. But they will all, without exception, have one process that carries information away from the neuron. One motor output for each cell, and those are called axons. The axons are the pieces of the neuron that carry information away. So this is a beautiful example of cerebral cortex, these little teardrop or pyramid-shaped cells, and I'm going to show you that again here. Give you a little bonus material that's not in your textbook about nervous system histology. Your teacher has done a lot of it in his life, so to give you a couple of examples as to what these things look like in different parts of the body. The other cell type that is in the nervous system are what are called support cells, or glial cells, or sometimes neuroglial cells. The word glia means glue, and so in the anatomist imagination, these are the cells that hold it all together, the glue in the nervous system. As it turns out, we know now that there are several varieties of these glial cells. So I want to show you a smattering of this. Again, we'll do this in the nervous system, and this is certainly not an exhaustive list, but you can see I've got oligodendrocyte, an ependymal cell, a microglial cell, astrocytes, all listed here as glial cells. There are different kinds of glial cells, this nerve glue cells, and they do different things that are important in the nervous system. So again, we'll bring those up in time, and I will tell you all about them and what they are for in the nervous system. But I just wanted you to see with your own eyes today, these are support cells, and they have different functions. The neuron is the conductive cell, delivering information. The glial cell does other stuff. It myelinates. It moves CSF. It supports the blood-brain barrier. It serves as immunity. So they have different functions, the glial cells, and we'll cover them in the nervous system. All right, here's some histosections to help you see. And you're supposed to pick up a few other key introductory factoids here about the nervous system. Things that you can never forget. Okay, so the first thing you see here at the top, it says these are cells of the spinal cord. So this gives your teacher an opportunity to show you a beautiful neuron in the spinal cord, but to say a couple of fundamental things that you should never forget. Okay, so this is a motor neuron found in the spinal cord. It's a very famous picture here of this motor neuron. Some of the most beautiful motor neurons in all of the body are found here in the spinal cord. And anytime I see a picture like this with a beautiful neuron pictured like this, I know immediately where they took this section, because they're all located in one place. All these beautiful motor neurons are in one place. So if you look at a spinal cord section, you'll have in the center of the spinal cord section darker looking material than on the edges. We call that gray matter in the center of the spinal cord. And we will learn much more about this. We know why that tissue is gray and white. The white material that we see in the nervous system is axons. And the reason it's lighter in color is because the axons are all covered with insulation called myelin, and it gives the tissue the appearance of white. You can see a lot of that white around here in this tissue, but if you look at a spinal cord section or a brain section, that's going to be the first thing that jumps out at you. Some of it looks dark and some of it looks light, and you're going to remember today, the lighter material is insulated axon bundles, myelinated, insulated axon bundles. The darker areas are areas where myelin is less dense, where there are cell bodies that don't have the insulator myelin on them. So that's why they have gray. That's why they look darker in appearance. Okay? So as I look at my spinal cord section, I notice that the central parts are more gray and the outer parts are more light. So I know something fundamental about the nervous system just bringing this up. It's this. If signals are going to travel along highways, going to my brain and away from my brain, where will those roadways be in the spinal cord? In the center or around the edges? The highways that carry information in my spinal cord are all in the edges. Where are the neurons that collect that information and deliver that information? And the center parts, the neuron cell bodies. Okay, so this is one of those locations, and it is the most famous of all of them. This is a neuron of the ventral horn of the gray matter of the spinal cord. A famous group of neurons known as the LMNs, the lower motor neurons. And that's one of them right there. That is a lower motor neuron. And I know, because I am in the ventral horn of the spinal cord in a lower motor neuron, that the information leaving this neuron is going to make a muscle contract. If I'm on Miss Frizzle's school bus and I get on board this neuron and I ride this axon out, it will take me to a skeletal muscle. So our job in this class is going to be to get on the school bus and follow these axon bundles out to their muscles. Where does this thing go? I want to track that axon. That's a lower motor neuron in the spinal cord, in the ventral horn. A skeletal muscle controlling neuron. All right, here's another very famous section. A broad view here and a little closer view. This is in the example. These are always used in the example slides. You can see here neurons all over this. This is the cerebral cortex. So it's dominated. You can see here that this is a highly concentrated area of neuronal cell bodies. What will that look like to you? If I look at the brain and I see a highly concentrated area of cell bodies, what will be the general appearance of that? Will it be gray or white? It'll be gray. Yeah. So in the brain, the arrangement is a little bit different. So this is the cerebral cortex. This is the outside part of the brain. So when you look at your brain tissue, you will see the outer part of the brain is gray. That's where the densest concentration of cell bodies are. So when we learn cerebral cortex, the outer part of the brain, every introductory picture of cortex has got a picture of the brain and these arrows pointing to all these different regions all over the cortex ascribing function. So the functional zones of the human brain are cortical on the outsides. And the neurons that dominate those areas that you can see here look like, can you see the teardrops? Can you guys see the teardrops? They are described by anatomists as pyramid-shaped neurons or pyramidal neurons. Now, I'm not saying anything in these introductory lectures that I don't expect you to know for this test and every other exam that's coming. What do you mean pyramidal neurons? Well, let me give you context. As we begin to learn pathways to muscles, the major motor pathway to skeletal muscles in your body is going to begin in the cerebral cortex on these neurons. And they're pyramid-shaped or teardrop-shaped. So we're going to call those neurons pyramidal neurons, and we're going to call that pathway, thousands of pathways, pyramidal tracks. This is part of the reason why they're called pyramidal tracks, because they begin in the cerebral cortex with these pyramidal-shaped neurons. Now there's another reason, and I think anatomists would give you a couple of these, so I'll bring them both up. There's another reason why they're called pyramidal tracks. And that is because if you get on board the axon here and you track it down, it's going to pass through, that axon's going to pass through your brain stem. And on the front side of your brain stem, at the level just before it gets to the spinal cord, there are these two pyramid-shaped structures that stick off of your brain stem. And all of the axons that are going to skeletal muscles that come from these neurons will pass through that area. Pyramids. So there are at least two reasons why we call them pyramidal tracks. One, because the neurons themselves, the original neurons in the cortex, are pyramidal shaped. And the second reason is the brainstem structures through which the axons pass are also pyramid-shaped structures. So, very famous, yes? Pyramidal neurons, pyramidal tracks are the motor pathways to skeletal muscles. We will begin those explorations in the cerebral cortex. All right? This is another brain area, very famous brain section here of the cerebellum. So, without this being labeled, this is just a low magnification view of cerebellum with a Nissl stain on it. And if you look at this stain here of the cerebellum, you go, I know immediately what that is. There's nothing else in the brain or the nervous system that looks like that. It's a very famous picture of cerebellum. And you can clearly see here a lighter area and a darker area. So, you're anatomy students now. Why is this area darker than this area?
Neurons and axons, different areas.
Right. It's a denser area of neuronal cell bodies, the darker tissue. Exactly. So the cerebellum has a very famous arrangement of layers. The cerebellum is always learned as a three-layered system. The outer layer here you can see is called the molecular layer. The middle layer is called the Purkinje cell layer, and this darker layer inside is called the granular cell layer. The statistics on the connections of axonal endings that terminate and leave granular cells are numbers that, I'll give you some of those later. They are staggering. in terms of the complexity. Thousands of connections per cell and billions of cells. I mean, it's the connections are, the number of connections within this structure are mind-boggling. You can't process. You know, the kind of connections in terms of numbers that rival the number of total molecules in the universe. It's the numbers are beyond imagination in terms of complexity. There will never be a computer that can do this. The guy who was my advisor when I was a graduate student, now at the University of Kentucky, but he was an Aggie for a while, and I would sit in front of the computer screen a lot, and it bothered me when things didn't work out. And so we were studying animal behavior, and I would look at the rhythmic behavior of an animal, and we were using all kinds of statistics to analyze those, rhythm statistics to look for patterns in the data. And the software I had would oftentimes not see the patterns, but I could look at the data and see the patterns. And so I'd say, Dr. Casson, and I would show him the patterns, and look at that. I see the patterns there. But the Fourier transform is not picking them up. And he would say, Wade, you need to use the big computer. Yeah? I mean, these computers that we use are small fry in terms of connection compared to what this computer does. So let's use the big computer. The connections are fantastically complex.